who is Dr. Erwin van Wijk, and he did his PhD at the Radboud UMC here in Nijmegen, the Netherlands. And he's currently an assistant professor and group leader at the otorhinolaryngology department, or in short, ENT. And his research focuses on Usher syndrome. And for that, his group is working on disease modeling using zebrafish, amongst other models, which it turned to be a very useful model for Usher syndrome. And at the same time, his group also focuses on the development of therapeutic approaches for different subtypes of Usher syndrome. And today he's going to particularly talk about exon-skipping for USH2A. So Erwin, the floor is yours.
Thank you very much for your kind introduction, Alex, and also for the invitation to present my data here on this wonderful platform. So today I'm going to talk about splice modulation therapy for Usher syndrome type 2A associated retinal degeneration. But first start with explaining what Usher syndrome is. Well, Usher syndrome is a recessively inherited disorder named after the Scottish ophthalmologist Charles Howard Usher, already in 1935 after he gave his Bauman lecture. And the syndrome is both genetically and clinically heterogeneous. So far, 10 different genes and mutations in these genes have been identified. And these mutations in these genes act via a so-called loss of function disease mechanism. It's a rare disorder with a prevalence of 1 in 20,000 individuals. And the main clinical manifestations are hearing impairment from birth. And this hearing impairment can be compensated by providing patients with cochlear implants or hearing aids, and vision loss due to retinitis pigmentosa. And for this retinitis pigmentosa, no treatment is available yet. And occasionally also balance problems occur, so vestibular dysfunction, as a consequence of vestibular dysfunction. So as said, Usher syndrome is genetically heterogeneous. However, mutations in one single gene, which is the USH2A gene, are the most important cause of Usher syndrome, explaining approximately 50% of all Usher syndrome cases. And up to now, more than 600 individual mutations have been identified that can result in either Usher syndrome in approximately 170,000 people worldwide, but alternatively, also mutations in the same gene can result in non-syndromic retinitis pigmentosa in approximately 250,000 people worldwide. So looking at how this disease progresses, as said, Usher syndrome type 2A patients are born congenitally hearing impaired. And this hearing impairment can be partially rescued by providing them with hearing aids. And at early adolescence, the first symptoms of RP become apparent, which are often a sign of night blindness. And this night blindness progresses into a form of legal blindness at an age of approximately 50 to 60 years, which leaves in between a window of opportunity in which we therapeutically can intervene in order to prevent or delay the symptoms. So when talking about therapeutic solutions, two aspects are of key importance. First of all, you would need to develop a therapeutic strategy. And second of all, you need to have a functional model in which you can assess this therapeutic strategy, whether or not it works. And as a functional model, as said, we use the zebrafish. Well, the zebrafish is a tropical freshwater fish that has been introduced to the laboratories already back in the 70s by George Freisinger in Oregon. And looking at the genome of the fish, we see that the USH2A gene is present as a single copy. And both gene and protein structure between human and zebrafish USH2A are extremely highly conserved, making this, for us, a suitable model to start with. So by adopting the programmable nuclease CRISPR-Cas9, and thereby relying on the non-homologous end joining repair pathway, we were able to introduce a net 4 base pair deletion, so a frame shift, in exon 13 of the zebrafish USH2A gene, resulting in a premature stop of protein translation and a null allele. We recorded ERGs in larvae of these knockout fish, homozygous larvae, already at an age of 5 days post-fertilization. And we were able to show that the B wave amplitudes of the ERG were significantly decreased in these fish, implicating that these fish are indeed already at an age of 5 days, are vision impaired. So making this a very suitable and useful model for our further studies. So the options for treatment, as said, mutations act via a so-called loss of function disease mechanism. So gene augmentation could be a suitable option. However, looking specifically at the USH2A gene and also other Usher genes, this is true. The size of the coding sequence is extremely large and by far exceeds the current capacities of the viral vehicles for gene delivery. And also, for multiple Usher genes, several isoforms are present. So which one to augment, that's the question. And also, when using gene augmentation, you really have to take into account that expression levels of genes are often critical, which are also difficult to regulate. Alternatively, we could think of gene correction strategies. However, also risks of off-target editing are still present, and these correction strategies are mutation specific. And as I said, already in the USH2A gene, over 600 individual mutations have been identified, making this not particularly suitable. Also, the efficiency for gene correction is currently not very high, and also immune reactivity against the Cas9 protein is still a potential problem. Also, mechanism-based treatments you could think of. However, the exact biological mechanism underlying Usher syndrome is still not completely known, so making this also a difficult approach. So ideally, we would like to interfere at the level of RNA, thereby not altering the protein isoform landscape in the retina and also not altering gene expression levels. And we choose the solution splice modulation for that, using antisense oligonucleotides that are complementary to the pre-messenger RNA, and upon binding, preventing the binding of splice factors, thereby inducing the skipping of exons during the process of splicing. And for AONs, we have two flavors. First of all, using AONs, you could correct aberrantly spliced exons or pseudoexons. And we've done that for a frequent deep intronic mutation in USH2A gene, resulting in the inclusion of pseudoexon 40. A second approach of application of antisense oligonucleotides is the skipping of native in-frame exons that contain frequent mutations. And this is where the rest of my talk will be about today. So 600 individual mutations are present that are all rare in the USH2A gene. However, 2 further mutations are present in the exact same exon, and it's exon 13. Normally, mRNA is being translated in protein with a fully functional, which is fully functional. And in case of mutations in exon 13, a premature stop codon is being introduced, resulting in a total loss of protein function. Exon 13 is an in-frame exon, so skipping of this exon would result in a restoration of the open reading frame, resulting in a slightly shortened protein with hopefully residual function. So that's the basics. If we perform in silico predictions, we do see that exon 13 encodes part of the 10 EGF-LAM domains that are present in the N-terminal region of the protein. And skipping of exon 13 would result in the loss of EGF-LAM domains 5, 6, and 7, and a fusion of EGF-LAM domain 4 and 8 into a functionally similar EGF-like domain. So how to assess then the effect of this on the level of visual function? Well, as I have shown, we have a zebrafish model with a truncating mutation in exon 13, so we could use that model for that. So to obtain proof of concept for our idea, we ask ourselves 2 questions. First of all, are we able to specifically induce the skipping of exon 13 in this zebrafish model? And we used a trial and error design for that, and test after that the most potent AON or combination of AONs that were capable of skipping exon 13. And second of all is there any protein made upon skipping of exon 13? And if so, is it present at the proper subcellular location in the retina? And does it result in improvement of ERG traces? So to start with the first part, we were indeed able to identify a combination of two AONs that were able to specifically induce the skipping of exon 13 in zebrafish. And if you look at the upper, or at this fragment over here, it seems that we were able to do that quite efficiently. However, upon quantification, we noticed that approximately 22% of the original transcripts were indeed the transcripts that we were looking for, so without exon 13, and that only 5% of so-called wild type, so untargeted transcripts were left, meaning that we approximately 70% of transcripts were lost, either naturally or upon targeting with these antisense oligonucleotides. So it's only 22% of the anticipated transcripts were there. After staining the retina of treated larvae with an antibody directed against the C-terminal end of the USH2A protein, we noticed that indeed part of the protein was again made, which was not present in our mutants. And upon quantification, we noticed that we observed approximately 20% increase as compared to the RMC, and also 20% of the original amount of wild type usherin was produced. So that was pretty well in line with the amount of transcripts that we noticed in our zebrafish that were treated with AONs. ERG recordings revealed that apparently this 20% of protein increase was enough to completely restore ERG traces in our RMC1 mutants, as compared to control treated and untreated RMC1 fish. So, meaning that we could conclude in the end that our mutants that we generated showed the absence of the USH2A protein, and already at 5 dpf, an early retinal dysfunction that we could record. And that the skipping of this exon 13 resulted in a partial return of USH2A expression, but that it had a correct subcellular distribution as anticipated. And that this 20% of protein that was produced was enough to restore ERG traces. So we concluded from that exon 13 skipping was a promising therapeutic approach for the future treatment of USH2A associated retinal degeneration caused by mutations in exon 13. So the next step was to translate these findings into a use in man. So for that, we performed a small AON walk. In this case, we displayed here 14 different antisense oligonucleotides that all had a phosphorothioate backbone with the 2 MOA modification. And we transfected these AONs in cultured retinoblastoma viri cells that endogenously express the USH2A gene. And on the left side, you see the untreated cells. And what we notice there is that you do see a transcript, which we here depict as being USH2A full length, but it is in PCR amplification from exon 10 to exon 15. And what we also see is that you observe a natural skipping of exons 12 and 13 in these cells, and also a natural skipping of exons 12 to 14 in part of the transcripts. Upon treatment of our AON, we eventually choose, in this case, number 5 as our lead molecule, which was named QR421A afterwards. Upon treatment, we did not observe any remaining so-called full-length transcripts left. We did observe a significant production of delta exon 13 transcripts, and also a small reduction in the delta 12-13 transcripts. So making this our ideal candidate for the study. So we performed a small dose series in transfection in patient-derived IPSC photoreceptor precursor cells. And what we noticed is that upon transfection, we did see a dose-dependent increase of exon 13 skipping as anticipated. And it is quite effective. Upon transfection at the maximum dose that we used here, 200 nanomolars, we do see a percentage of approximately 60% of transcripts that lack exon 13. However, if we would translate this to the retina, we obviously not going to perform a transfection there, but we perform a naked delivery into the vitreous. So we model this by a gymnotic delivery of our AON to the medium of IPSC-derived photoreceptor precursor cells. And also here we do see a dose-dependent increase. However, the dose or the amount of skipping that we noticed went up to approximately 17.5 to 18%, quite matching the amount of skipping that we observed in our zebrafish model. So we designed a surrogate QR421A molecule from mouse, and we injected this into the vitreous of wild-type mouse model. And using a fluorescent probe, we looked at the distribution of this AON upon a single intravitreal injection. And we noticed that after 7 days, this AON was distributed through all the layers of the retina, including the RPE, and that this persisted until day 259, and there we stopped looking, but still at day 259 we could detect this AON in all different layers of the retina, meaning that it's apparently quite stable. Also, as it is adjusted to the mouse sequence, we noticed that exon 13 skipping was indeed effectively induced. So we performed also a dose response using different doses after a single injection, and watched after or harvested these eyes after 7 days. And we did see an increase in the amount of exon skipping, although it caps at approximately a dose of 30 micrograms per eye. So we used that dose and followed that up in time. And that's plot D. And notice that if we inject this 30 micrograms dose, it peaks at an approximately 50% of exon skipped transcripts, and then it's quite stable. But still at day 200, after a single intravitreal dose, still a level of 40% of delta exon 13 transcripts was seen. So after some safety measurements, ProQR Therapeutics, with whom we collaborated, decided to launch a clinical phase 1/2 clinical trial, and the first patients were dosed in March 2019, which was quite something, as this was the very first clinical trial for the treatment of USH2A associated retinal dysfunction, almost mainly based on the proof of concept that we obtained solely in zebrafish without the interference of an alternative larger animal model. So last April, the positive intermediate results were reported, meaning that QR421A is well tolerated and no severe adverse events and inflammation was observed in any of the patients. And this molecule demonstrated benefit in several parameters that we included of vision, including best corrected visual acuity and static perimetry and retinal imaging. And the most interesting part is that this treatment benefited all treated individuals, including patients that were already in an advanced stage. And that was not something that we anticipated on beforehand. And what we notice is that the best corrected visual acuity will be the parameter that will be used as an outcome measure in the next phase 2/3 trial in the advanced population, as it was seen that on average, patients had a 9.3 letter benefit in the treated eye as compared to the untreated eye in this patient group. And in the early to moderate population that was included, static perimetry will be the outcome measure, measuring the retinal sensitivity. And also there, we did see a significant increase in the amount of loci with more than 7 dB increase. And what you see here, and that's quite interesting, after 12 weeks after the initial single treatment, you do see that the effect wears off, meaning that we probably have to re-administer somewhere in between the 12 and the 24 weeks again in this population. And as said, due to these positive outcome, two pivotal clinical phase 2-3 clinical trials are being prepared right now. One to also test this in advanced population, which is named the Sirius trial, and one in the early to moderate population, which will be named the Celeste trial. And with that, I would like to thank you and thank all the collaborators and all the funding bodies that have made all this possible. And I'm open to any questions that might pop up. Thank you very much.
Thank you very much, Erwin. So I have a lot of questions, but there are already some in the chat. So I will start for those ones, and because I can always ask you the other questions in person. So you mentioned that the clinical trial, oh, now the chat moved. You mentioned that the clinical trial started without going through an animal species larger than a zebrafish. So what about toxicity studies?
Toxicity, that's a good question. Toxicity studies have been done in an in vitro test. That's one. Second of all, well, I have shown you that we have obviously looked at the distribution of this and also the kinetics of this molecule in mouse models. But the real proof of concept, and that's what the point that I would like to make, the proof of concept that this really works, so that it restores visual function, and then it's, the remaining protein is indeed functional. That is only based on experiments in zebrafish. So we obviously did use other models for showing other things, including toxicity, but the proof of concept that it works was only derived from zebrafish.
Okay, thank you. Frida, would you mind to stop sharing, please, so we can see each other? And then, well, I had a question over the 10% that it's enough. So do you think that this is something related to the maybe the half-life of USH2A protein, or it's something that can be extended or expanded to other proteins of the retina? Well,
that's obviously a very interesting question. We do know that the half-life time of USH2A, it's a large protein, so it takes quite some time to generate this protein. We also know that it's not extremely highly expressed, and we do know that it's quite stable. So probably that what you indicate is true. So if this will be the case for all other molecules and all other targets, well, that we can only speculate about that, but I can imagine that there will be more proteins, including the larger ones that we are also struggling with. Other Usher molecules also behave, well, quite similar as this one. So, but I'm not sure whether or not we may extrapolate it to everything.
So the Junos also asked, what does RMC1 stands for as a mutation type?
Oh, sorry. Yeah, I should have mentioned that. It is just a nomenclature that we have to that we get from the Zebrafish International Resource Center, because we have to administer. We sent this mutation to a stock center where it's being registered, and RMC means Radboud Medical Center. And then it's the first mutant that we were able to generate within a Radboud zebrafish facility. So it's Radboud Medical Center, mutant number 1. So that was this line.
Okay. Then Gemma also asked about the periodicity of administration is probably related to the protein stability and half-life, but how many intravitreal injections are accepted by the eye without an adverse response?
Yeah, that's a good point. So now we anticipate, based on the half-life time and of this molecule and the effect, that we need to re-administer approximately, well, let's say 3 times a year, 2 to 3 times a year. That's what we anticipate. For other well, disorders, we also need to re-administer, well, other drugs multiple times a year. So upon intravitreal injection, and without any, well, real disadvantage effects. We do not know whether or not, if we would do this for years, if this would result in any problems with application of AONs. However, so far as I know, and as far is known in literature, multiple dosing of these AONs, well, does not lead to major problems. However, this will probably over time, when more information becomes available and re-administration will be done also for other conditions, like the one you are also working on, Alex. I think we will learn more from that in the near future.